Okay, I think when Dan made the comment in the last session that a lot of social science genomics is not even really about biology and genetics, it's about using those tools to answer social questions that are of general interest to social scientists. I think that's a good sort of lead into what I want to talk about. I'm just going to show a bunch of different studies that are in various stages of completion, some completed, some with a lot of revision ahead, to give you a flavor of what some of us in sociology are doing with these genetic information. Dan kind of already covered, I didn't know in the intro you were going to actually get into the causal model, but he mentioned the summative score, the polygenic score, where you sum over all those individual alleles, and then you have some decent explanatory power that you would not have, especially for social science relevant phenotypes, if you were focused on individual alleles. So I'm going to skip that, but you're going to talk endlessly, I'm sure, about polygenic scores over the course of these 2 weeks. But I'm sure many of you have seen this chart already from the paper last year that James was the first author on, where the group developed a polygenic score for educational attainment that had an out-of-sample incremental R squared of about 12 to 14%, depending on what data set you were predicting in. And to give a sense of what that means, if you look at the blue bars, which, if I remember, are add health, and you're in the bottom quintile of the score distribution, you're predicted to have a 12% chance of graduating with a four-year college degree. And if you're in the top quintile, you're predicted to have almost a 60% chance of graduating with a four-year college degree in the United States. So a five-fold difference. So they're a long way away from individual prediction, but if you bin people into groups, you're going to predict their group level average education pretty well. So you can imagine a lot of applications for that I'll talk about at the end. So I'll go through a bunch of topics: so family life, race and ethnicity, peer effects, health, kind of in rapid succession, not necessarily in that order. We'll start with families. So one thing you can do with these polygenic scores is ask how alike are mates. Now, this is different than genetic assortative mating on the allele by allele. Because if I have a polygenic score that's at the mean, and my partner has a polygenic score at the mean, we could be actually very different on our individual alleles. We could be almost uncorrelated at all, but we got to that mean score. So we got to that mean score in different ways, but we're correlated at that mean score. So it's not when we see these correlations, for example, in education, we know that there's a high degree of assortative mating in education. The left lighter bars in this chart are phenotypic correlations between spouses in the health and retirement study, and the right darker bars are the genotypic or the polygenic score correlations. And you see that, for example, education, as many demographers know, demonstrates a high level of phenotypic correlation between spouses. I think second only to political belief these days, although we don't have any political scientists anymore, right? So no one's going to correct me if that's not true. But when you look at the genome-wide level, you see that it's about less than about a quarter of the strength of the association and phenotype. Now, we don't think when we talk about a polygenic score correlation between spouses, it's not some magical pheromonal process by which these alleles are attracting each other through odor, which there are some theories about attraction and odor, although maybe that is a very small part of it. We're talking about phenotypic processes happening where people are sorting in the educational system and in the marriage market, and that drags along the the alleles for a ride, and then that's why you get this observed genetic correlation. That said, the consequences of it are important for demographers because we know that to the degree that there's assortative mating on a given trait, particularly at the genetic level, then the variance for the next generation should increase. And that will have important consequences for inequality in education, for example. When we look at height, it's actually an interesting case, the only case where the genotypic correlation is higher than the phenotypic correlation. And that's probably because height is, the genetics of height is actually a proxy for a lot of health-related manifest phenotypes that are maybe too subtle to measure. Body symmetry and other sort of developmental indicators. BMI and depression, those are poor polygenic scores. They're among the poorest in this group, but there's no genetic correlation. There's a robust phenotypic correlation, but of course, this is not longitudinal data. I don't have sort of a population and following who marries whom and who doesn't get married and so forth. So it's very possible, and this is measured late in life, so it's very possible that, that the phenotypic correlation is entirely through not assortative mating, but mutual influence. Your wife is making you depressed. If your wife is depressed, believe me, you're going to be depressed, and vice versa. Or you're eating the same food at home for 20 years, your BMI is going to be correlated. So we don't know if that's assortative mating or not. But at the genetic level, as I mentioned, the importance is because that affects the variance of the next generation. If we look at differential fertility, that's going to affect the mean level of the next generation's phenotype. So again, since the early 20th century in the US and at various points in history in other countries, the positive association between education level or social class more generally and fertility that some people, for example, the economist Gregory Clark argued was part of the explanation for why the Industrial Revolution started in the UK, was a differential fertility among the upper and lower classes, flips with the demographic revolution and gets more and more strongly negative as time progresses. And in the HRS sample, we can see that education is about negative 0.15 correlated with number of children ever born. But when we look at the actual genetic correlation, which is about, is our population going to have more, on average, more education enhancing alleles or education detracting alleles, assuming the environment is constant and what alleles matter are the same 30 years from now, we see that it's much smaller, but there's still a negative correlation at the PGS level between education, PGS, and number ever born. Again, height acts weirdly, opposite directions for phenotype and genotype. So I'm best guessing that's something nonlinear going on there, that very tall people have low fertility, but in general, the genetics of height, as I mentioned before, are a sign of and promote healthy phenotypes in terms of developmental indicators, and therefore they're positively associated with fertility. Parenting. An alumna of this program, actually, Asta Breitholt, and I'm probably pronouncing her name incorrectly, and I looked in ALSPAC, which has very detailed measures of parenting for very young children, to see if parents were responsive to the child's polygenic score, which of course at birth, absent, this is prior to, to parents like getting their baby saliva and sending it in immediately to 23andMe or something, but it is not an observable trait, it's not an observable datum for children. Their sort of cognitive promise unfolds over time, and parents respond to that. And as Dan mentioned, a lot of the pathways are through the environment. So part of the educational attainment polygenic score works through what we call evocative RGE, meaning the kids with a higher polygenic score evoke a differential parental response than the kids with a lower polygenic score. In other words, when it's time for bed, the kid with the higher polygenic score is probably asking for one more book or two more books before bed, and the kid with lower polygenic score is most likely not kind of evoking that same response, that cognitively stimulating response from their parents. That's at least the theory. So we looked in 3 samples. Sample 1 is a very big sample, but it has the kid's polygenic score, and if I remember correctly, the mom's just the kid and a bunch of control variables. Sample 2 has the kid's polygenic score and the mom's polygenic score, and sample 3 is the gold standard sample, which is we have both parents' polygenic scores, so the kid is a random draw. So if the parents are at 50th percentile, the kid could be a draw at 40th percentile or at 60th percentile, and how do the parents react to that? Now, it's a much smaller sample because a lot of the dads were not genotyped. Dads are harder to get a hold of course. So we show all 3 to show that the kind of substantively they're the same. So you see at 6 months, there is no effect of the kid's polygenic score. Maybe there's just not enough sort of, wow, what can they do at 6 months to show me that they're like a genius? I don't know. They're laughing more or they're babbling. Maybe they pay attention a little longer if you show them a book. I don't know, but there doesn't seem to be a baseline response at 6 months. But as you go to older ages, up to 56 months, you see that kids with higher polygenic scores for education evoke higher, more cognitively stimulating parental responses. Our preferred sample crosses 0, the confidence interval, but if you see the kind of general pattern, the point estimates are all positive and around the same magnitude. So that's work in progress. You'll hear a lot about genetic nurture. I think, Dan, you already mentioned it, or someone in the audience when they were talking about the threats to the sort of the causal model. We're talking about genetic nurturance, which is the fact that 50% of your alleles are shared with your parents. So they're having a causal effect through the parents' behavior and therefore structuring your environment, like just talked about the different parenting strategies, for example. Likewise, if you have siblings, you're sharing on average, not deterministically like with parents and children, but on average 50% of your genome, and they're part of your social environment too. So among fraternal twins in the Minnesota Twin Family Study, we looked at how does your siblings' polygenic score affect your attainment. And you see that this is the respondent's polygenic score effect. So this is in standardized units. Every additional standard deviation of polygenic score leads to about a 2/3 standard deviation increase in educational attainment. But the siblings' polygenic score has a sizable effect as well at 0.4 standard deviation, 0.4 elasticity. We looked at for an interaction effect, and we didn't find one. But the interesting part for us was that when we broke it out, these were fraternal same-sex fraternal siblings. There's no cross-sex pairs in the data, unfortunately. But when we look at sisters versus brothers, we found that sisters have a big effect on, and are what driving this sibling effect in the total sample, and brothers have 0 effect on each other. Which again, this is using the genetics to tell us something about social relationships in the family, which have generally been a black box for social scientists. It's very hard to disentangle causal relationships within the family. Up till now, the only kind of random assignment that many of us have been able to use is the sex of the firstborn child, for example, which in the West we can treat as randomly assigned. But now we have something else, again, conditional on parents' genotype or a family fixed effect that is randomly assigned, and we can then, that can be a mechanism to sort of unpack some social dynamics within the family. I'm only here till tomorrow, so I apologize that I can't stay longer. I have a 2-month-old, and also apologies for any errors I make from lack of sleep because of that. So I'm happy to talk during the breaks, or you can email me. See, now from the 2-month-old, I forgot even my train of thought what I was going to say. He'll come back to me.
Could I ask a quick question?
Yeah.
I was actually wondering, so in terms of sort of the confounding factors, I was wondering if you could actually talk a little bit about that. So none of this is sort of longitudinal data in terms of socioeconomic status changes or anything like that, right? This is all sort of single time point.
Right. Well, the ALSPAC is longitudinal, but
so could you just give a sense of like how do you correct for things like socioeconomic status?
well, if you again, this goes back to what Dan was saying. If you have that ideal study design where you have everybody's genome in the family, then there's no confounding with the socioeconomic status because it's conditional. It's a random draw. I mean, many of us who went into this, I don't know what this is called, subfield or whatever, new field, were driven by the notion that in order to properly account for, explain human behavior, it's ridiculous to ignore the environmental side. It's equally ridiculous to ignore the genetic side, the biological side, given we know from 50 years of twin studies, I don't know if David's going to talk about this, that the sort of median heritability for traits was 0.49, and maybe a little lower on average for behavioral, but a significant chunk, and that many of us went into this interested in not only getting a complete picture or controlling for the genetics so we can we get less biased estimates of the social environment, but ultimately, and I'll get to this later on, gene-environment interaction, where things like changes to income come into play more for that kind of thing. But since it's fixed over the life course, that's sort of, we're getting the average effect across all those states. You got to speak up because I'm very hard. The message? The message is, again, this goes in the bin of very preliminary, but that it seems as though there are more, and this is one data set, very small sample, and we want to dig into this, but there seems to be indicator that sisters affect each other more than brothers in terms of their educational progression in the family. Whether that's through competition, whether that's through collaboration, as like Carol Gilligan would have us believe, we don't know, but it's something to explore more. So another thing that is helpful for, again, because it's fixed at birth, and solves a lot of problems of reverse causation, is understanding peer effects. So here we see that, for example, among educational, for the educational polygenic score, there's a correlation in Ad Health, the data that you're going to use, of 0.11 among friends. Remember, it was 0.15 for spouses, so it's pretty, people are sorting among their friends almost as much as they're sorting among who they marry genetically. If you control for schools, if you look within schools, so a lot of that correlation could just be class-based differences of where families live. But so you look within schools, it gets cut to about half, but it's still significant, marginally significant. We see no effect on height, and if we look at a body mass index, we find that the correlation in body mass index among American adolescents is is unexplained by differences across schools. It's entirely within school phenomenon. So that's just that's not social genetic effects. That's saying friends are correlated on their genome. It's obviously, they're not making each other's genome more similar. So that's a threat to the idea that you could just look at correlations between friends and assume that's a causal effect of peers on each other. So that's the problem of homophily. Another problem in peer research is what Charles Mansky calls the reflection problem. If Dan and I both go into sociogenomics, we don't know whether it was Dan influencing me or I was influencing Dan. How do we know if if we both are smokers and it's not homophily, who influenced who? Unless, I don't know, maybe there's some big data, like you could get such granular data you'll be able to tell in our minds who was influencing who. That's a general problem. And then there's the contextual effect. Are Dan and I both smoking because we're stressed out that we're having to teach at this summer institute and like the projector's not working? So that's not a peer effect, that's a context effect. So how do we get rid of all those? So we use an identification strategy. We look at adolescent smoking, and we use school fixed effects. So we're identifying off of, and we use the polygenic score for smoking among the individuals in a school, and so we know it's fixed at birth. There's no my smoking behavior is not affecting Dan's polygenic score. We could still be friends because we both have polygenic scores that predispose us to smoking. So we don't emphasize sort of the correlation among friends. Instead, we use the school fixed effects and we identify off of the grade differences. So Dan and I are in 11th grade, and there happens to be in our school, 11th grade has, because of, we're going to argue, random variation across years, happens to have a higher mean level of smoking polygenic score risk than the 10th grade, or the people who are in 11th grade next year. If everyone follows that. So assuming there's no gentrification happening specifically, there's a trend in the polygenic scores within a school and so forth, then we show that. We argue that this is as if random, and we can identify the effect of your grade mates on your own behavior. If that all makes sense. And the polygenic score is key because we don't have to look at the actual behavior that could be changing, could be a reflection effect. It's unchanging, and therefore we know it's influencing ego rather than ego influencing alter. So when we do that, and I would say our preferred specification is the reduced form of just the mean polygenic score for the grade. We also looked at bad apples and shining lights models. So bad apples is that some values at extreme of the distribution of high risk for smoking are what driving the effect, and shining light is that people at extremely low genetic propensity of smoking is what's driving the effect. It turns out that bad apples is what's going on, which makes a lot of sense for something like smoking. Maybe for academic achievement, shining lights would be better. Anyway, as a check, we look at height, and we have very noisy zeros for height. Some of you who study peer effects or networks might remember Jason Fletcher's paper that kind of ridiculed or made fun of Nicholas Christakis' JAMA paper that claimed that obesity spread through networks by showing that height, acne, headaches, I think that was maybe one more, spread through social networks as well. In other words, showing that what Christakis and Fowler were showing was basically just homophily, that people with the same BMI were friends, because there's clearly no causal effect of your friends on your height. So just to give you a sense, it's obviously a much noisier estimate because it's a weak predictor, but I mean, it's got a lot of measurement error, but it's at least if you even look at the left end of the confidence interval, the peer genetic environment seems to have a pretty big effect, on the order of being male or even your own polygenic score. And what's, again, that is a tool then to understand social dynamics in American schools. So we're underpowered to really get these differences, but I think it's at least suggestive or interesting to see that if you look at the biggest point estimates, of course these are overlapping, but is for white respondents responding to the polygenic scores of white grade mates. And if you look at all grade mates, the effect of course, the confidence interval gets smaller because of the bigger sample size, but the effect gets smaller. I would love to have a much, 10 times the sample size to really identify these effects, but it tells you that white, at least suggestive that it could be possible that white peers are only responsive or predominantly responsive to other white students' behaviors and not to minority students' behaviors. Give you another example of where we're using genetics, but we're really using it for a social or a social epidemiological story. There's been a lot of work suggesting that within the African American community, those with darker skin have higher rates of social stress, experience discrimination, and therefore the negative health consequences of that, specifically hypertension. Of course, just merely looking at a cross-section of the population and looking for a correlation between darker skin and hypertension could be driven by any number of things. For example, the average skin tone in the American South is darker than it is in the North among African Americans, and the South for blacks and whites is known as the stroke belt because of very poor health behaviors, diet, and so forth. So could be driven by region, it could be driven by poverty. We don't know what's going on. So we take, again, Add Health, which has a skin tone measure and has siblings, and we use the sibling fixed effects model, and we find that the lighter skin sibling has a lower risk of being diagnosed with hypertension, and we also look at systolic and diastolic blood pressure, and you see that we controlled for time outdoors and so forth. It's a significant effect within families. But you have a question?
Yeah. So these are full siblings?
Yes.
Yeah, but it is within the European population, right?
No, this is African Americans only.
But then it doesn't have the genetic aspect.
Just hold on a second. So there's no genetics here yet,
Okay.
right? This is just sibling. So we're already, I think, moving the ball forward to say it's not just a mere correlation, because within families we're seeing that randomly assigned, based on genetics, skin tone differences between siblings is driving hypertension. It still could be that there's genetic pleiotropy, which you'll hear a lot about this couple of weeks, that the same alleles that affect skin tone have a direct effect on cardiovascular health. One thing you'll learn is that there's no single gene that has one function. So even one of the classic ones, alcohol dehydrogenase, is expressed in many different tissues other than the liver and has many different functions other than detoxifying you after a night out. So it's very possible that it could be that this is just a genetic effect. It's not a social discrimination effect. And just as a side note, because I'm not going to be here when it happens, I'm very skeptical of genetic instrumental variables or Mendelian randomization. Dan and I had this ongoing kind of discussion for years, and I'm happy to discuss that in the downtime. But because of this fundamental pleiotropy of genes, it's very hard to meet the exclusion restriction. that is necessary for Mendelian randomization, especially with social and behavioral traits, where so much of the causal mechanism is working through selecting into environments and social interaction and so forth. So anyway, what we do instead is that we looked at the 8 at the time, the top 8 SNPs that determine skin tone and eye color, and see among the non-black population whether they have an effect, just a reduced form effect on hypertension. And we found one that was significant, but it goes the wrong direction. In other words, lighter coloration has a higher rate of hypertension. So we're able to use some genetic information. Now, this is not comprehensive, again, to sort of chip away at that alternative explanation that it's genes that's driving the skin tone hypertension relationship. So, as I mentioned before, many of us who go into this, or at least the sociologists, are really motivated by the notion that, you know, if there is a gene for aggression or whatever, a genotype for aggression, and you're born into a impoverished, you know, neighborhood in West Baltimore, that genotype is going to lead you to not good things, like probably, you know, violence and jail. But if you have the same exact genotype and you're born into, you know, Cranbrook, you go to Cranbrook Academy where Mitt Romney went, it's going to lead you to be that innate aggressivity is going to lead you to good things, probably, you know, to being a corporate raider or venture capitalist or whatever. This idea that GE is really fundamental to our understanding of social behavior and the interaction between the role of biology and environment. Another way I like to think about it is heterogeneous treatment effects are rife in the social science. If you have a randomized trial for reading, improvement of reading, and Dan's in the treatment group and he gets some special new phonics or whatever whole word approach of intensive reading tutoring, and we measure his score before the start of the year, and we measure mine. I'm in the control group, or this half of the room is in the control group, and this half of the room is in the treatment group, and we see that there's a 10% increase in the treatment group. That's great. That 10% is a pretty big treatment effect for those kind of interventions. But when you look at the data, you'll probably see that that 10% is driven by the fact that Dan is now 10% of the kids in the treatment group double their reading scores, and there maybe was no effect for the other 90% or something. There's heterogeneous responses to randomized treatments are kind of rife. And so the notion is, at least the intuition is that this is another area where genotype can step in and help us make sense of heterogeneous treatment effects. That's a role that biology is having. So the problem is, and if it's hard enough to get sort of data where you have random assignment of G, because you need the family structure data, like I guess Alex is coming next week, right? We had access to Decode Iceland, where you basically have everybody in the family and can really identify effects, or sibling fixed effects that you have, which is kind of second best choice, that you have in Ad Health that you're going to be using. It's hard to get it's hard enough to get G to be exogenous. Then, as all applied labor economists in here know, E is incredibly hard as well to find a natural experiment or do a randomized trial where you're going to actually be confident that the environmental exposure is not correlated with genotype. And then finding that in the same data is, next to impossible. So I could say, I could count on my, probably 2 hands the number of human gene environment interaction studies that I believe. A third issue I'm not going to get into today is how do we measure genotype for gene environment interactions? So The examples I'll show are using a standard polygenic score. But think about what the standard polygenic score is. It's usually from a meta-analysis. At the very least, it's in one data set. The discovery of the effect sizes is from one data set where there's many birth cohorts involved, say like the HRS. Most likely, it's a combination of UK Biobank, 23andMe, HRS, all the sort of scraps that went together to get 1.1 million individuals from different countries, from different birth cohorts, from different socioeconomic statuses, which suggests that the signal that you're getting out of that is the genetic signal that is robust or sort of unaltered by that wide range of social environments that you're estimating on. So perhaps we're missing a lot of gene-environment interaction by using regular polygenic scores. So another approach is to use that we're working on is to use polygenic scores that predict variation rather than levels, and then interact those with the environment. But that's not what I'm going to talk about today. I'll just give a classic example. Economists in the room know that the original Angus paper, which actually was copied from a sociologist, Norman Hurst, who published in the New England Journal of Medicine, looking at the mortality effects right after the Vietnam War. But Angus looked at the labor market impact of military service during the Vietnam era and used the draft lottery as a natural experiment. We follow in that tradition and use the draft lottery, and we interact it with the smoking polygenic score, and we see that this is joint work with Lauren Schmitz, who will be here again next week, I think, to talk about epigenetics, who is also a graduate of this, isn't she? To show that if you are basically have a protective genotype against smoking, you're not likely to be addicted to cigarettes. There's no treatment effect of being drafted during Vietnam, but if you already at genetic risk, there appears to be a sizable effect on your lifetime smoking behavior, which of course then translates to other health risks. I don't mean to steal your thunder or anything, but it's such a great example. I wanted to show Patrick's paper that uses a schooling reform in the UK and looks at its impact on body mass index. And overall, there's an impact on the distribution of body mass index. But if you look by polygenic score tercile, and whether they use the polygenic score for BMI itself or the polygenic score for education, you see that prior to the reform, there's more spread among the polygenic scores in terms of the phenotype. In other words, there's a bigger effect of the polygenic score on BMI, whether you're using the BMI polygenic score or even more so by education. And then after the schooling reform, that's compressed. So there's a gene-environment interaction. Did I misrepresent that at all?
Yeah, I mean, it's a funny gene, because this is the policy and not the effect of the policy. But sure.
Okay. Anyway, it's very, on one hand, it's very hard to find the right data to do where you have genes, you have a big enough sample size to do an IV or regression discontinuity and so forth. But you could imagine a lot of existing strategies like a schooling reform RD or a draft lottery instrumental variable and adding a genetic component to it as more and more data become available. Okay, how much time do I have left?
10 minutes.
10 minutes, okay. So I'm going to quickly talk about another area, issue of race, which I should say that with the exception of the school smoking study, which uses all respondents, and in fact smoking is one of the phenotypes for which there is pretty good prediction for non-white populations in existing polygenic scores. All of the results I've presented so far are based on white non-Hispanic respondents, or those of European descent. In the UK, it's called white British. US, it's called white non-Hispanic. And that's for a whole variety of reasons. Alicia, I think, is going to talk, where is she? Is going to talk about sort of portability issues or polygenic scores. But there's a sort of subtext here that in this very research we're creating or reifying or something racial inequalities by that very sort of, for those very technical reasons of needing same ancestry populations in your discovery and in your replication samples. And since there's just currently a lot more European data available, that's a big issue. So I have only 10 minutes. I'm going to skip this to say that there a whole area of research that I think sociologists and social scientists in general have not really got that much into and is really ripe for investigation is using the genome as a historical record. So, for example, if you look at black-white intermarriage in the United States, or intermating, you'll see that in the two pulses of admixture that occurred, one in the antebellum South and one in the post-Civil War South, there's an overrepresentation of European DNA in the Y chromosome. In other words, that tells us that there is the social process that occurred for mixed race offspring was that there was white fathers disproportionately and African descent mothers. And we know that historically from rape and of slaves and so forth. That flips in more recent times, where we know from just general census data that the most typical, I think that in terms of black-white marriages that result in children, 3 times as many are from black men and white women than the reverse. So using genetic data across, filling in the gap between 1860 and today would tell us a lot about the social history by looking simply at the ratio of European and African ancestry in the Y chromosome versus the autosomes. So that's just one example of how you can use, instead of punting on race, you can actually use the information in the genome to try to understand social processes in historical time, recent history. You could also look for, I don't know if you can see this well, but the black dots are self-identified African Americans, the gray dots are self-identified whites, and there is a zone where some of the African Americans have more European ancestry, because on the x-axis left is European and right is African, and some of the whites have more African ancestry. And so, interesting question is, when there's a disjuncture between self-identified race and ancestry, is that a stressful condition? Is that passing? Does that create health issues or stress? You had a question?
Yeah, actually. So speaking of genetic components, because some of these studies, especially about the history using the genome as historic record, sparked some controversies, especially because quantitative measures were used without any consultation with race scholars, for example, on how to define race, whether there is genetic basis of race or not. And most race scholars firmly say there's no genetic basis. And it seemed always that quantitative geneticists were kind of stepping into the backyard of others and dominating the narrative. So, to what extent do you think race scholars should be involved in framing the narrative rather than just being consulted distantly if they were consulted? And another thing is, do you think these narratives, do you think genetics would fundamentally help these narratives, or let the historians do their job and maybe we can assist them partially?
Can I answer the second one first? Yeah. So the second one, I think that this is from Sarah Tishkoff's paper, and I think it helps to use the quantitative genetic data. It helps the narrative because you can see, for example, that the distance between a Fulani herder and a Nilo-Saharan African is about as far as a European and an East Asian. So that sort of it helps deconstruct the common sense notions of race by using genetic distance to show that they're pretty arbitrary. On the other hand, a lot of scholars are very uncomfortable, race scholars are very uncomfortable with using genetic ancestry at all. I mean, do others want to say something about that? It's definitely, it's kind of a catch-22 because I think a lot of scholars have focused on, look, we're not going to deal with race. It's such a hot-button topic. It has a very troubled history in terms of the intersection with genetics. So we're going to just stick with this UK biobank, white British, and just run our analysis. But of course, that itself is creating, if there's anything good that comes out of all this analysis for healthcare or precision medicine or what have you, it's creating a health care disparity by sticking to that strategy. So I don't know the answer. I know that people are trying to engage with race scholars, but unfortunately, I think if anything, it's gotten more polarized lately for a variety of reasons, like the reporting of what's her name in the New York Times, who was at our conference. Amy Harmon, yeah, who through her journalism basically linked the work of scholars and white supremacists, doing us no favors to be able to just quietly do work without addressing, without getting embroiled in that kind of crap. But yeah, so if anything, I think it's very hard to find kind of common ground where scholars from these different camps can talk about collaboration but that's just my very anecdotal sense, so I don't know.
I would say a critical point is that race is not the same thing as genetic ancestry for all sorts of reasons, including that race is a social construct that is defined differently in different societies. What it means to be black in the U.S. Is different than what it means to be black in Brazil, for example. So I think it's important to understand that these measures, although correlated with race, are not measures of race. And also, in terms of communication, I think it's important to avoid referring to these genetic measures as measures of race or measures of ethnicity. Refer to them as measures of ancestry, because that is what they are, and I think it helps avoid reifying race, which I think is problematic.
Yeah, an important issue. I think we should keep discussing it over the course of the 2 weeks. Okay, so I just want to say we talk about the PGS. It's a really great tool. Depending on what you're using it for, it's got some problems. I mean, of course, Alicia's going to talk a lot about the portability issue of why you can't just take a polygenic score estimated on white British and apply it to people of Asian or African descent, for example. But there's some kind of quirkiness going on even within Ancestry. So this is, again, white British UK biobank only. And you can see that when a GWAS the discovery is done among all respondents, and then the prediction is done within quartiles of SES, you see that it predicts better the education for low SES individuals. than it does for high SES. That's not itself that surprising. Eric Turkheimer and others have actually argued the opposite, that there's a environmental suppression of genetic effects in low SES using twin models, and that you basically see that high SES parents have the resources to allow the sort of genetics of each child to reach their full potential. So you see a stronger genetic effect in high SES. This says the opposite, but it's even more interesting or weird is that when the GWAS is estimated in among the low SES sample, its predictive power declines across the quartile. It's better, as we saw, but it declines across the SES quartile. But when it's estimated in the high SES group, it's consistent across the quartile, although lower. So we really don't know, what's going on exactly, but it suggests that not just population stratification, not just allele frequency differences or LD structure, linkage disequilibrium, but G by E and these social genetic effects, the genetic nurturance, very much varies by social circumstance, and that's something that has to be taken into consideration. Likewise, if you look at the polygenic score where the betas are estimated in the traditional way, just throw a big unrelated sample in the pot and estimate the betas, versus when it's estimated based on SIB regression, the sort of gold standard we were talking about. You see that for something like hair color, so this is like adding more SNPs to the polygenic score, where you go from just genome-wide really big hits to all SNPs. You see that something like hair color obviously tracks pretty well, but when we get to social outcomes, you see, first of all, for years of schooling, there's a gap between the prediction, and this is exact same sample size, between the predictive ability of the SIB-GWAS, which is lower, and gets rid of social genetic effects like the genetic nurturance and so forth, versus the traditional GWAS. But even something for like height. Okay, is that? Oh, you weren't pulling the plug on me literally at the time. Even something like height, as you get further into the smaller and smaller effects, relaxing the p-value threshold, then do we have to turn that on again or something?
There's a power button on the top.
You see that there's a divergence. Okay, I'm gonna, I know I'm out of time, right? So I just wanna, am I?
Oh, yeah, you're right. But you should finish.
So another thing that we're looking at is sort of the social consequences of polygenic score prediction. This is, as Dan was mentioning, the price is falling faster than Moore's law in microcomputing. So the amount of data, I think latest count, it was like 26 million Americans, almost 1 in 10 have participated in some form of recreational genetics or genomics, like Ancestry.com or 23andMe. So increasingly, Americans and worldwide, people are going to have access to their own genetic data. There's very little just straight social science scholarship on the implications and how people view the use of these data. They're potentially could be used by insurance companies. It's outlawed in the United States for health insurance and hiring from GINA, but car insurance, long-term care insurance, life insurance, all, I'm just surprised that the companies aren't already asking for your 23andMe login. Because some of them are asking for you to wear like John Hancock asked for you to wear a fitness tracker. They start by just saying, we'll give you a discount if you share that, but then it becomes mandatory. And whether that's something we want or we want to fight, I don't know, but because yes, they're risk adjusting and kind of defeating the purpose of insurance. On the other hand, if they're not allowed to do that, then you have adverse selection as more and more Americans will, oh my God, look, I have like much higher than average probability of getting Alzheimer's, I'm going to buy long-term care insurance, and then you can get a death spiral if you don't let the insurance companies risk adjust. So you can imagine if you're from LA or New York, how crazy school admissions are. I mean, I know you're not at that stage yet, but there's like consultants for 4-year-olds to get into schools, so forth. You could imagine there's an incredible measurement error for trying to assess the cognitive potential of a 4-year-old, but you could imagine these schools requesting polygenic scores to go along with the application. You could imagine, we don't have to imagine anymore because Steve Su and others have created a startup where IVF, couples using IVF select embryos based on their polygenic scores for implantation. All these things are unfolding rapidly, and we know little about, maybe Americans are fine with all that. I don't know. The UAE, other countries are going full steam ahead with these kind of things as well. So that's something else we're looking at by doing a national representative sample survey of attitudes towards the use of polygenic scores. So they're really an important social issue, and social scientists should be engaged even if they're not interested in causal estimates of altering an allele at conception or whatever. So I'll stop there. Thanks.